Hello class. So this lecture is on nucleotides and nucleic acids. So as far as nucleotides, there is three basic components to it. It's one, the nitrogenous base, two, a pentose, which is a carbohydrate, and then the last but not least, one or more phosphates. So as far as the nitrogenous base, there are two derivatives, which is purine and pyrimidine. And so the purine has two rings. One ring is a six-member ring with two nitrogen groups that is adjoined to a five-member ring with two nitrogen. So that is purine. And then pyrimidine is just one six-member ring with two nitrogen groups. And then as you can see right here where these N has been labeled, that is what is linked to the sugar, which is the pentose, and has that glycosidic bond that I mentioned in the previous lecture. And then as far as the structure for these nucleotide bases, we have for the purines, you have adenine and guanine. And then for as far as the pyrimidines, you have cytosine, thymine, which is basically in DNA, and then you have uracil that is in RNA. And then know the difference between thymine and uracil is that methyl group. So a methyl group is with the thymine, and with it removed is the uracil as a nucleotide base. And then the second component, which is the carbohydrate, which is the pentose. And another name for this pentose is ribose. And so when you have ribose, you have on the second position, you have the hydroxyl group. But when the oxygen is removed, then you have what is called a deoxyribose. This is found in DNA, and the ribose is found in RNA. And then at the end, you have an ester bond for that third component, where you have the phosphate group, where you can have one or more phosphate group attached. And then you have this nucleotide all together, the phosphate group, the pentose, or the carbohydrate group, and then you have your base, where not is the purine base or the pyrimidine base. And then as far as the sugar, the ribonucleotide has the hydroxyl group on the second carbon of the carbohydrate ring, and then when you have the oxygen removed, this is the deoxyribonucleotide. And then as far as the nomenclature for a ribonucleotide, you need to know the structures, name, numbering, and symbols, such as the one letter and three letter codes. And so right here you have an example of adenylate, which has a one letter of A and then AMP. And then this M, this stands for mono, then P is phosphate, so there's only one phosphate group. And then the A is correlated to the actual name of the base. And then we know that with a ribonucleotide, once again on that second carbon of the carbohydrate, you have a hydroxyl group. And then so on and so forth for the rest of the bases, and notate that there is no thymine base here because this is for RNA, and so that is where you have the uracil type of base. And what is highlighted here, that's kind of in red or this peachy type of color, it is highlighting what is called a nucleosine. And so a nucleosine has a base and the carbohydrate. The only thing it's missing is the phosphate group. So when you have the phosphate group, it's a nucleotide, which is right here. And then when the phosphate group is missing, you have a nucleoside. And they have different naming nomenclature as well. You have adenosine, guanosine, and uridine, and cytidine. And then as far as the nomenclature for a deoxyribonucleotides, which is found in DNA, you need to know the structures, names, numbering, and symbols, both two-letter. Notice that there is a D that is placed right in front of the letter, and that's the major difference between the ribonucleotides and the deoxyribonucleotide. And then as far as the four-letter, once again you have that D that is ahead of the normal three letters that we see with the ribonucleotide. And everything else is pretty much the same as far as definition, but notice that now we don't have that uracil. We have the thymine that is here because that's correlated with DNA. And you still have, when you just have the base and the carbohydrates, that's a nucleoside. And with the phosphate group added to it, that makes it a nucleotide. And then here is some more naming nomenclatures with it. So when you have just the base, you have for purines, you have the INE, so adenine, guanine, and then when you go into pyrimidine, you can still have the ending of INE right here, but the only one that's different is the uracil. And then when you come to having nucleoside, with the nucleoside, with the purines, it has an OS than INE for purines, but the pyrimidine has the ID in front, and that's replacing that OS, and it still has the same last three letters of the INE. And so that's how you kind of know the difference between the two types of bases with a nucleoside. Same thing with a nucleotide. You have an ending of a YLATE. And then with a pyrimidine, you have the ID that is placed in front to have for the naming nomenclature. And this is just more so correlating what goes for what, whether or not you're dealing with a ribonucleotide or ribonucleic acid or a deoxynucleic acid. And this is just the same type of chart. It's just a little bit more cleaner for you to reference. And so as far as the third component, the phosphate group, that is our energy source. And so once again, when you remember when I mentioned the AMP, what that MP stands for a monophosphate group. That's when you have this one phosphate group. But when you have two of them, it's ADP. So, and then when you have three of them, it's ATP, which ATP is very much well known. So, starting from the ribose, the three phosphates are generally labeled alpha, beta, and gamma. Hydrolysis of nucleoside triphosphates provides the chemical energy to drive many cellular reactions. Adenosine 5-triphosphate, which is also called for short ATP, is by far the most widely used nucleoside triphosphate for this purpose, but UTP, GTP, and CTP are also used in some reactions. And so as far as the role of nucleic nucleotides. The most well-known, once again, is ATP, but as far as roles for nucleotides, is energy-rich, which includes not only ATP, but GTP, CTP, and UTP. Nucleotides can also act as carrier molecules, dealing with NADH and FAD. We'll definitely be talking about within Biochemistry 1, especially when we go into glycolysis. As far as UTP sugars, I think that is more so in Biochemistry Part 2 from my memory, but we'll see as the semester progresses. And then you have secondary messengers, and then finally other cofactors for enzymatic type of activity. And here are some of the structures of other nucleotides that we may be studying. And then as far as joining together nucleotides is a phosphodiester bonds, which is the backbone of alternating pentose and phosphate groups in both types of nucleic acid, is highly polar. The 5 and 3 end of the macromolecule may be free or may have an attached phosphoryl group. And so when you see with DNA having that 5 to 3, that 5 is associated with the carbon that is with the pentose ring. And so remember, you start with that alphanumeric carbons. That's 1, 2, 3, 4, 5. So on that fifth position, you have that phosphate group. So that's why you have a 5 end, and then the other attachment of the phosphate group is on the third carbon of the carbohydrate ring, and from there it attached to the fifth carbon, and then another attachment on the third carbon attaching to the fifth carbon, and then on, at least for this picture, you have a free macromolecule for that third position on this carbohydrate. And so as far as DNA, it has 2 strands of nucleic acids that are bond together using hydrogen bonds, and then you have these base pairings. So you have adenine that is base paired with thymine, and then guanine is base paired with cytosine. The discovery of the structure of DNA by Watson and Crick in 1953 gave rise to entirely new disciplines. So regardless of the species, the number of adenosine residues is equal to the number of thymidine residues, and the number of guanosine residues is equal to the number of cytidine residues. Thus, having this equation, A plus G is equal to T plus C, and this relationship is called the Chargaff rules. And then as far as the stability of DNA can denature by having acid and base around that puts charges on the bases, which causes internal repulsions repulsions and break up the double bond helix. And then denaturization can also be accomplished using heat. And then you'll see, as far as this picture, this random coral is being, for this double helix, is being unwound and thus denatured. And so the roles of nucleic acids is information storage, retrieval, translation, process, and preservation. So we'll go more and more in the details of that later on. Just kind of have this fundamental knowledge for right now. And then last but not least, I know you're probably very familiar with DNA having a double helix, but DNA can actually have a triplex. And this is called the Hooke's pairing, which allows this formation. And so what happens is what you see for right here. You have a cytidine, and then right here, and then you have guanosine, and then you have cytosine as well, which is shown right here, and you have two pyrimidine rings, and then you have one purine, which is right here. And so not only can you have a triplex, but you can also have 4 DNA strands that's paired together, making a tetraplex. But this occurs readily only by DNA sequences with a high proportion of guanosine residues. And that concludes the lecture on nucleotides and nucleic acids.